Welcome back, all of you, to another session. The next speaker we have with us is Dr. Abhrajyoti Ghosh. Dr. Abhrajyoti Ghosh is a distinguished researcher at Bose Institute, specializing in microbial stress response and adaptation. His work mainly focuses on understanding the molecular mechanisms that allow microorganisms to survive and adapt to challenging environments. Dr. Ghosh employs a variety of techniques, including biochemistry, microbiology, and genomics, to identify and characterize the key molecular players involved in microbial survival. His research also extends to studying the diversity, distribution, and function of microorganisms in unique ecosystems such as the Sundarban mangrove and Darjeeling tea plantations, using genomic and proteomic approaches to discover novel metabolic pathways. Dr. Ghosh brings a wealth of expertise and insight to this FDP program, offering participants valuable knowledge and perspectives on today. His topic is basically biological advances in remediation of heavy metal, a direction of resilient agriculture. Over to you, sir. We welcome you and we are really honored to have you with us today.
Thank you. Thank you. Am I audible?
Yes, sir, you're audible.
Okay, so should I share my slide? Share screen. Choose screen. Wait. Share Windows. Windows. System is blocking screen share due to denied permission. Allow. Wait. Can you see my slides?
No, sir, not yet.
Okay. Wait.
Professor Ghosh,
Yeah.
can I share my screen to show you how to do that?
Yeah, you can do.
Okay.
I'm having issues.
Can you see my screen?
Yeah, I can see your screen.
Okay.
Should I share the slides with you?
So
Maybe.
yeah, at first you can do one thing. So you can see that leave option.
Yeah.
So just behind that, there is this logo that is called sharing.
Yeah, I know.
So if you do that, so then it will take you to two options. One is called screen and one is background.
Yes, I know.
So if you open the screen, so then the entire screen will be displayed.
I know.
Then you can, and then you can open your PPT.
Choose share this
If
screen.
it works in that way, it's fine. If not, then we can share on behalf of you.
Yeah, I think.
Yeah, we can see something is coming out.
Okay.
Yeah, we can see the thank you slides. Yeah.
Now, can you see?
Yeah, we can see, but it's very hazy, Payal.
Yes, sir, it is a little hazy.
It's very easy. We
Hazy?
can't see it properly.
Okay.
Slides are being displayed. That's not an issue, but the main problem is the readability index. It's very hazy.
Okay. Let me try. Now is it better?
Not yet, not yet, not yet. You can do one thing. You can send your PPT to us and we can do it from our end.
Also, I can do one thing. I can change the computer. Just wait. It will take
No
5
problem.
minutes.
If you want, we can also do that.
Yeah, I'm changing my computer. No issue.
Okay.
Just give me 5 minutes.
No problem, no problem, no problem.
Something wrong in this.
Yes, sir, it is fine now.
It is fine.
Yes, sir, clear.
Oh, I think I have already changed the computer. So what I will do, I will use another computer. That's better, I think.
Okay, sir.
Just 2 minutes. I'm already done.
Okay, sir.
I'm joining to that. Hello. Please allow me once again.
Yes.
Yes. Okay. Yes. Can you now? I can share now, I guess. Enter screen. Share. Now, am I? Can you see the slides?
Sir, it is fine.
Okay. Okay. Sorry for the delay. So, thank you for the introduction and for inviting me to give this lecture. So, So today I will be talking about the biotechnological advances in remediation of heavy metal, a direction of resilient agriculture.
So you're not audible. Your screen is visible, but you're not audible.
I am not audible. Why? But it's showing it's fine. Uh, am I audible now?
It's fine. Now we can hear you.
Okay.
Yes, sir, you are audible as well as slides are visible.
Uh, okay. So So I'm from Bose Institute, Kolkata. So in my lab, we are working on microbial stress response and adaptation in general. So we have two kind of research going on in our lab. So in one group, we are working on model organisms, how these model organisms actually adapt to different kinds of stresses and show resilience to changing environment. On the other hand, our group is also interested in looking at microbes in a community perspective. So that's where today's talk lies mostly. So, so normally, if you think about a microbe, it does not live alone in the environment. Rather, it lives with many other microbes in natural environment, constituting microbial community. Now, this microbial community actually face a lot of different kinds of environmental challenges in the form of different kinds of stress. For example, thermal stress, oxidative stress, nutrient stress, solvent stress, and most importantly, nowadays we hear the term called anthropogenic stress. So basically, these are human-made pollutants that create a lot of stress to different kinds of microbial community. We are particularly interested in heavy metals, potentially toxic elements. So nowadays, heavy metals are also called as potentially toxic elements because the terminology of heavy metal is not really acceptable because many of the non-heavy metals are also showing toxicity. So I will be talking about mostly the potentially toxic elements. Now, if you want to understand the effect of anthropogenic stress on microbial community, you need to have ecosystems. So you need model ecosystems. So we are particularly working with two different kinds of very contrasting ecosystems. One is the tea ecosystem of the Darjeeling and surrounding areas, where we are trying to understand how the chemical fertilizers and also different kinds of agrochemicals are contributing towards the changes in microbial community and dynamics. On the other hand, we are also looking at the effect of anthropogenic stress on the microbial community of the mangrove ecosystem that we have in the southern part of West Bengal. It's in the Sundarban area. So today I will be talking about the mangrove ecosystem. So mangroves are the top tropical environments. They are situated in the intersection of land and sea. They are having sediments that are mostly anoxic and reductive, mostly because of regular inundation that you all know how it works. And despite of these environments are very diverse and rich in animal, not only animal species but also different plant species. Now we will be discussing about Sundarban, which is situated in the northeastern coast of India. It is the largest mangrove delta of the world, and it is a habitat of rare and endangered animals. Mangrove Tigerland, the only mangrove tigerland in the world. It is World Heritage Site and it has unique species richness and diversity that I will be touching upon. Now, where comes the microbe? Because I am mostly interested in microbes, and it has been shown by many people, many researchers, that microorganisms are integral part of any mangrove ecosystem. Now the question comes, what they do in mangrove ecosystem. So they contribute towards biogeochemical cycling of different kinds of nutrients. For example, they help in recycling and transforming various nutrients and thus make the mangrove ecosystem more productive. At this point, I should mention that mangrove ecosystems are very productive ecosystems considering many other ecosystems around us, and they have a resilience because of such productivity. Now, the second thing that microorganisms actually contribute is in conservation and rehabilitation of mangrove ecosystem. So there are microbes that contribute towards biocimenting. That is one of the very important aspects when you talk about mangrove ecosystem. So sustainability and the contribution of microbes in that is one of the important features in case of mangrove ecosystem. And finally, bioremediation, where microorganisms are involved in a variety of biodegradation and remediation processes in mangrove ecosystem. And that is where we are mostly interested in to tap whether we can have some interesting microbe that can help us in developing some bioformulation for biomanufacturing purposes. So we started our work long back, more than 10 years now. So we asked three very basic questions when we started our work. So the first question was, who are there? So when we started in Sundarban mangrove, we didn't have much data available on what are the different kinds of microbes present and how they are contributing towards the maintenance of the ecosystem or how they can be exploited. So who are there is one of the very basic questions that we started our work with. Then what are they doing? They are present in that ecosystem, but what is that they are doing is very important to know and understand. And that was our second question. And finally, how do they survive and interact? So in natural environment, they are, as I already mentioned, they are not alone. They form community, but again the community isn't enough. The community interacts with different kinds of biotic and abiotic factors. So abiotic factors, I will be talking about the stresses, but also the biotic factors, for example, the mangrove plants and through rhizosphere, through endosphere. So they do interact, how that interaction actually offers survival of this microbial community and selection of those microbial community, and also how this microbial community interacts. What is the basis of crosstalk? What is the molecular mechanism of crosstalk between the community of microbes and the plants that are present in the mangrove ecosystem in Sundarban? Are some of the very basic questions that we started working with. Now, to address each of these questions, we actually started adapting different types of techniques. Uh, so the first, to address who are there, we adopted the technique called metagenomics. The term was coined by Professor Joe Handelsman in 1998. Nowadays, everybody is doing metagenomics. So we also started long back, more than 10 years back, to do some basics of metagenomics. And then we moved on. What are they doing to address this question? We went for isolation, characterization, functional gene diversity. At this point, I should mention that in metagenomics, we do look at microbes that are not culturable. So normally, among all the microbes in any given environment, 1 to 3% are culturable. So it is important to understand the holistic view of the microbes that are present in any environment, and metagenomics can be a good choice of study through which you can identify or look at the microbial diversity, irrespective of whether they are culturable or non-culturable. But when it comes down to what are they doing, often you have to go for isolation, characterization, and also functional gene diversity. So to address the second question, we move to the base very fundamental study where we isolated, characterized multiple organisms, and the basis on which we started isolating and characterizing microbes, that is one of the things that I am going to tell you today. And finally, to address the last question, how do they survive and interact? So that is what we are presently working on, but here we are mostly working on the functional genomics. So after isolating the organism, so we are trying to perform functional genomics to understand how these microbes are actually coping with anthropogenic stresses and also contributing towards bioremediation, for example. Let's move on. So Sundarban mangrove wetland, as I already mentioned, an UNESCO World Heritage Site. So this is situated in the northeastern coast of India. And from our lab and also from other lab, uh, results have shown that we have a defined gradient of pollutants, concentration gradient of pollutants in Sundarban, where near the inhabited area we have high load of pollutants, whereas near the sea or away from the inhabited area, we do see that we have a gradual decrease of the concentration of those pollutants, the same pollutants. So this is what we have been looking at, measuring in different studies in our lab, and also there are other labs who are also working in the similar area. And we all do see that, yes, indeed, we have a gradient of anthropogenic pollutants across Sundarban. Now, coming to potentially toxic elements, as I already mentioned, nowadays the heavy metal terminology is not really uh, accepted because a lot of non-heavy metals are also contributing towards pollution. And it's always important to understand the sources of all these potentially toxic elements. So there are multiple sources from which we have anthropogenic deposition of these potentially toxic elements in the mangrove environment. And those potentially toxic elements either convert into different kinds of salts, here you can see some reduction occurs for certain metals, for sometimes it is carbonated, and also sometimes it is oxidized. So these different forms of potentially toxic elements then eventually adsorb on the surface of the sediment particle, so the clay particle, and eventually reside there for very long time. This was the very fundamental aspect that we uh, looked into during our course of study. Now, so when we started working with, we actually were trying to make a catalog of microbes that are present in Sundarban. And the first hint of influence of this potentially toxic element obtained from a study back in 2015, where we were looking at uh, archaeal diversity. So archaea represent third domain of life uh, other than bacteria and eukaryotes. So they are very sensitive. They are very sensitive towards pollution. Only a subset of archaea can deal with a lot of pollutants, for example, heavy metals or potentially toxic elements, and also different organic compounds like poly aromatic hydrocarbons. So these subset of organisms can deal with that, but the rest cannot. So when we did this study, what we did see that in some of the uh, sediment samples that we analyzed, we did see that there are certain group of archaea that are highly enriched. These green ones are the enriched ones, whereas in some samples, we do see that other subset of archaeal organisms are heavily enriched. So when we performed analysis of heavy metals, polyaromatic hydrocarbons, so on and so forth, all the physicochemical parameters, we did see that, yes, indeed, there is a difference. So, for example, in Godkhali or Bonicam. So here we actually analyze samples collected from 3 different stations: Godkhali, Bonicam, and Dhulivashani. Dhulivashani is somewhere near the river mouth, which is near the, even beyond Kolosh, and which is very near to the sea. So there we do see we have a different archaeal community present compared to uh, in places where we have human intervention. For example, Gothkali and also Bonicam. So when we looked at the physicochemical parameters, how these physicochemical parameters are correlated with the samples and the organisms, archaeal organisms that we identified in our study, we did see certain things. For example, in Bonicamp and Godkhali, we did see that yes, they are highly correlated with different kinds of potentially toxic elements, as you can see over here: copper, zinc, cadmium, iron, so on and so forth, also lead over here. And not only that, a certain group of archaea, they are haloarchaea, basically halobacteriaceae family. So these haloarchaea are highly abundant in these sampling stations as well. Not only that, these sampling stations were heavily populated with different kinds of polyaromatic hydrocarbons. So when we went back to literature, what we found out that these haloarchaea are the only subset of archaea that can deal with heavy metal and polyaromatic hydrocarbons. So they have the metabolic pathways that can deal with heavy metals and polyaromatic hydrocarbon. Thereby, they are getting enriched in those sampling sites where we have high pollution load of polyaromatic hydrocarbon and potentially toxic element. So in that study, we actually proposed that haloarchaea as a proxies for hydrocarbon pollution as well as potentially toxic element pollution or heavy metal pollution. So that was the very fundamental study that we performed. Um, so in this particular study, if we think about it, was, so when we talk about metagenomics, there are different ways we can perform metagenomics. One way is we just target the 16S RNA gene. I believe all of you know. So 16S RNA gene, it's called amplicon sequencing based metagenomics. It's amplicon sequencing, basically what we call it now. So there we look at the 16S RNA gene, how many different kinds of 16S RNA genes are present in a given sample, and also how diverse they are. However, there are other techniques of metagenomics where we don't look at a single gene. Rather, what we do, we actually take out the entire soil DNA, cut it into pieces, and sequence all the pieces. And then we look at the diversity of microbes based on the rRNA gene that we do with amplicon sequencing as well. But in addition to that, we can also now look at other subset of genes that are present in the entire pool of DNA. So that is called whole genome metagenome sequencing. Now, lately, what we did, we actually moved from amplicon sequencing to whole genome metagenome analysis with Sundarban samples. And here the sampling sites were much more, distributed. So this is Gothkali over here. In between you have Bonicamp, and here you see it's Kolosh. Dhulivashan is somewhere near the Kolosh. But there were like 2 more sampling stations that we collected samples from. What we did, we collected soil samples from these environments, and then we performed all the physicochemical analysis of potential toxic elements, also analyze the uh, polyaromatic hydrocarbons and other organic molecules that are present in the samples. Along with that, we actually isolated the entire soil DNA or sediment DNA. Then we perform whole genome metagenome analysis. Now, when you have the entire data set, the first thing you ask, how diverse the microbes that are present in those sampling sites. The first thing we observe that there is a difference, and the difference is very striking. Why it is striking? We observe that pollutants lower the alpha diversity. Alpha diversity gives an indication of how the microbes are, how diverse is the microbial community in a given subset within two samples from a same environment, how different they are. So when we looked into the alpha diversity, we did see that pollutants lower the alpha diversity of resident microbiome, where we did see that Godkhali, which was the most polluted one, has the least diversity compared to, for example, Kolosh, where we have the least pollution, or as per our analysis of physicochemical data, we did see that we have the least pollution in Kolosh, and there the diversity is highest. That was really amazing to see. So more you have pollution, less you have the diversity of microbial community. Now when you have this data, the next thing you should go look at is the what are the dominant microbes in your population. So the first thing we observe that the microbiome or the microbial community is dominated by bacterial genera, and uh, this domination is always it's like the species like pseudomonas, streptomyces, burkholderia, they are really dominating the all the sites, and they are highly abundant in sites where we have more pollution. We also looked into the archaeal fraction. So in our previous study we showed the haloarchaea were dominant in different polluted sediments that we analyzed. And here also from the whole genome metagenome data, we did see, yes, indeed, if you go through this list, you see lot of haloorganisms. So they are highly abundant in sites where we have more pollution. This further supported our previous work where we showed that indeed haloarchaea is very sensitive. They can deal with these pollutants, and they are highly abundant in polluted sites of Sundarban. Now, as we have the whole genome metagenome data, you can look at other genes, let's say functional genes. So other than 16S rRNA gene, you can now look at other genes, metabolic genes that are present in those microbes or those DNA samples that you obtain from those sediments collected from Sundarban. When we looked at the diversity of functional genes in all these samples, we did see again the functional gene diversity is again lowest in case of Godkhali, where we have most pollution, and it is highest in case of Kolosh. Not only that, when we categorize these genes into different types, so you cannot go through this list, they are very small, but just to help you to understand, so these green subset of genes, these are enriched in highly polluted sites. And if you go through this list, which is you just have to believe my words, so these gene subset of gene contains a lot of genes that are enriched, and they are involved in heavy metal resistance and also mobilization, and also polyaromatic hydrocarbon degradation. We also identified certain genes that are depleted in low pass sites. So it's a low polluted sites, means they are eventually enriched in high polluted sites. So, and in this list of genes, we have lot of genes involved in heavy metal resistance, heavy metal mobilization, so on and so forth. So we concluded that, yeah, not only the diversity, but also not only the microbial diversity, but also functional gene diversity is getting affected by the pollution in Sundarban. Now, coming to the next thing, so we also looked at the antibiotic resistant genes, because we have the entire subset of functional genes in Sundarban, and when we looked at it, we did see that yes, indeed, we have high diversity. So in any natural environment, uh, antibiotic resistant genes are present. So any microbe, if you take out any microbe from natural environment, it has antibiotic resistant genes. But our objective was different. Our objective was to understand, if you increase pollution, whether you see changes in the abundance of a particular antibiotic resistant genes in any given environment. So we did see high diversity of antibiotic resistant genes in Sundarban. We compared our data with with data collected or already published from China and also from Kerala. And we actually could see that we have quite a lot of diversity of antibiotic resistant genes. So let me clarify here. So when you have more pollution, a lot of pollution, diversity of antibiotic resistant genes goes down. Means a particular group of antibiotic resistant genes goes very high, become highly abundant, and that creates a lot of nuisance in the entire ecosystem. So that's why we actually looked at it. But the funny thing was, when we compared all these data together, we did some statistics, and when we looked at their correlation, how the antibiotic resistant genes are really correlated, we did see that most of the samples are correlating well. However, there were like samples from Kerala and two samples from Shenzhen, China. They were like outliers. They didn't show any correlation with the rest of the data set. So our work was mostly concentrating on sediment samples, and we tried to include samples from, data from the sediment samples across the globe. And when we looked at this data, when we looked at these outliers, we were confused initially. And then eventually we realized that all the data that were published from Kerala were from rhizosphere, not from the sediment itself. And also, two samples were collected from rhizosphere from Shenzhen, allowing us to propose that, okay, in rhizosphere something else is happening. It's not the same like sediment. So that was the question when we moved to rhizosphere from the bulk sediment. Now, if we move on, we also started working with heavy metal resistant bacteria in two locally available plants, or most abundant plants in mangrove plants in Sundarban. One is Goran and the other one is Vine, Seriops decandra, the Goran, and Avicennia officinalis is the vine plants. You all possibly know. And we looked into the physicochemical parameters after collecting rhizosphere soil, and also we started analyzing the microbiome of the rhizosphere of both these plants. Now, when we performed some statistical analysis, we did see that Goran plants, especially, and in the polluted sediments, we do see there is a correlation, like Gotkali again, as you know, I already talked to you. So there are certain group of microbes that are highly correlated with the Goran, and also some of the heavy metals are also showing correlation with the sites from where we are actually we have collected the samples. So, but we do see that there is a distinct difference between the sediment microbiome and the rhizosphere microbiome. And we are going to propose actually that rhizosphere effect, which means the plant root exudates. So plant roots actually secrete exudates, and those exudates can modulate the microbiome of the rhizosphere. So we are proposing that rhizosphere effect possibly play an important role in shaping the rhizosphere microbiome in case of mangrove plant. So what we have seen so far, that when you have anthropogenic pollution, some organisms are selectively enriched in case of Goran plant, which is attributed by its root exudation as well. Whereas in case of vine, we didn't see much difference compared to the sediment. That is what we see. So there are influence that we could actually observe when we talk about rhizosphere microbiome and the sediment microbiome of Sundarban. Now, the next question is, well, we have organisms that are enriched in some of the mangrove rhizosphere. Can we culture some of these microbes from mangrove rhizosphere? So here comes our next story, where we actually looked into arsenic resistant bacteria that are halophilic. So in Sundarban, all organisms are halophiles. So halophilic arsenic resistant bacteria from the rhizosphere of Cereops decandra, Goran plant. And we ended up isolating, so we isolated more than 100 organisms, but eventually we came down to two organisms which showed the maximum potential. So one was Cucuria flava and the other one was Bacillus vietnamensis. And these two organisms are typical, reported previously in other studies, but we this is just a phylogenetic tree showing how they are related to uh, mm, the neighbors of the group. And also this is showing how good they are metabolically. They showed different kinds of metabolic activities. Uh, so they have some distinct activity compared to the other one, but they have some metabolic activity that are useful to survive in environment like Sundarban. Now, so to move on, we also looked into their. So this is the Cucuria flava and this is Bacillus vietnamensis. Though they both of them can form beautiful biofilm on last slide. Not only that, when we looked into as because they were isolated from the mangrove rhizosphere, so we tried to look into their ability to associate with the plant root. So this is the root of the rice plant grown in South 24 Parganas in high saline condition. You can see these organisms can form biofilm-like structure when I get associated with the root surface of the rice plants, giving the indication that yes, indeed they can get associated to with the root and possibly they can contribute towards the plant growth and development. So to check whether they can really can help in plant growth and development, we did some basic preliminary in vitro studies where we showed that they both these organisms can form indolacetic acid in absence and also in presence of tryptophan. So tryptophan biosynthesis is the pathway through which indolacetic acid is formed. So when you add extra tryptophan, you do see more indolacetic acid formation, but even in absence of tryptophan present in the media, you do see we have indolacetic acid formation. Also, both these organisms are highly potent in producing siderophores. Siderophores are really helping the organism and indirectly the plants to get uh, metals like iron in soluble form. So they can chelate iron and offer it to either plant or the microorganism. And both these organisms are extremely capable in showing resistance to arsenic. So the arsenite is the toxic one. Here you see we have the MIC value of 12 to 15 millimolar, whereas arsenic, which is less toxic, here we have even more resistance shown by these two organisms. So both organisms show promise in PGP activities. Now, what about in planta experiment? When we move to in planta, what we see that here you see this is a control experiment where if you inoculate without giving any stress to plant, you see in both inoculation, both these organisms can help showing plant growth promotion compared to control. Now, in each of these sets, we have different kinds of stresses. In the first one, we have increased arsenic. So it was 2 millimolar arsenic added to the soil right system where we could see that you see the control plant. It is really very sick. Whereas when you inoculated these two organisms, the plants are much more healthy and the root systems are better. Also, when we increase the salt concentration, we do see salt can really affect the growth of rice plant. It was done in IS64, but in case of inoculated, uh, I mean, experimental sets, we do see we have nice growth of the rice plants, even at higher salinity with uh, salinity sensitive variety of rice. When you have these two organisms, the plants or the rice plant seedlings can grow better compared to the control. Now we included another set where we had both salt and arsenic together, and there you see the control plant already died here. It already died, but when we had either of these inoculum added to the uh, system, the growth was quite nice for the seedling, allowing us to conclude that yes, indeed, these two organisms can help plant growth and development even at high salt concentration and at high in presence of arsenic. Now the next question comes in our mind, always in everybody's mind. It will come that what is the mechanism of arsenic resistance? Well, they can show arsenic resistance that we know from the in vitro studies. We also know in presence of arsenic, it can help plant to grow better compared to the control. But how they are doing it? That is the next question that we started asking. And when we asked this question, we actually did a lot of electron microscopy, and this is one of the team. This is just same EDUX analysis data. I don't want to go into the detail. This is a control cucuria, and here you see that if you look at this arsenic, you see there is no arsenic in the membrane. So one of the proposed theory in literature that they absorb heavy metals on their membrane structure. So we checked it, and when we checked it, we did see that indeed, when in absence of arsenic, there is no arsenic on the membrane structure when we analyzed the at the atomic level. But when we grew the cells in presence of arsenic, we did see the concentration, high concentration of arsenic in the membrane structure. So possibly they absorb arsenic on their membrane surface. And the same thing, same phenomena was observed when we looked into the Bacillus vietnamensis, and there also we did see, yes, indeed, arsenic is getting accumulated on their membrane surface. So we did further electron microscopy with TEMIDUX, where we actually sectioned the cells and looked into the arsenic concentration in different parts, and I can rest assured tell you that yes, indeed, we did show in this study that in the membrane structure, they actually modify the arsenic and keep them in a stage where the arsenic remains immobilized and not easily mobilized. And the effect I will show in shortly. So just to summarize that, well, these two organisms that we isolated, these were potential plant growth promoting organisms that can show promise in plant growth promotion at elevated salt concentration as well as at high concentration of heavy metal or potentially toxic element. Let me tell you, we also checked other heavy metals like cadmium, lead, mercury, and in every cases we did see that they have positive effect, and they show promise towards growth promotion, as I already mentioned, in presence of high salt and arsenic. Now what so far we have shown that arsenic adsorption is one of the key features that these two organisms are having, and then also by sectioning we showed that arsenic is getting accumulated in different membrane structure from further electron microscopy analysis. Also, we showed that root association and biofilm formation play a key role in getting these organisms to associate with the rice plants, and eventually by measuring the heavy metal concentration in different parts of the plant by using atomic absorption spectroscopy and other techniques as well, we showed that there is a reduction of arsenic accumulation and growth promotion simultaneously in the rice seedling when we have these two organisms inoculated or added to the soil right system. So in a nutshell, I would say that these two organisms, when you add them, what they do, they absorb arsenic even if they are present in the soil, then they accumulate arsenic within their membrane structure, thereby allowing less arsenic to go into the seedling through ascent of sap, and thereby allow less arsenic to get accumulated in different parts of plants, which eventually get accumulated by other studies. We now know that they eventually get accumulated in the seeds of the rice plants. So these or two organisms actually offered a possible bioremediation where we have arsenic reduction of arsenic concentration in rice plants. Now, the next thing is very important. So there are set of questions. Let me just briefly go to it. Is the water for irrigation contains high concentration of potentially toxic elements? So this is just a question. So normally, people have shown that the irrigation water that are available in 24 South 24 Parganas, they are having potentially toxic elements present, and it's more than the danger level in certain cases. But again, at the same time, we don't use saline water for irrigation. We don't use saline water at all for irrigation. So it has to be sweet water, sweet water bodies where from we use water for irrigation, but they have potentially toxic elements. Now, the next thing is very important. What happens to the heavy metals when sweet water is used for irrigation? Is there any other way that is helping? Because we know we have lot of cultivation going on in South 24 Parganas. So that's why we decided to move a little further from these studies, and we decided to check the heavy metals and microbiome in the irrigation water that are available in those areas. Let me show you. So these are some of the sweetwater pond systems in Chagor Island and Ghoramara Island, from where waters are used for irrigation and even for household uh, use. So yeah, so what what we did see that in these water bodies we have lot of sponges growing, so both in Chagor Island and in Koravara. So there are different species of sponge. They are really populating these water bodies, especially during summer, uh, and eventually they fade out, uh, and we were really interested. So when we looked at the heavy metal concentration in this water, we do see there is elevated concentration of heavy metal, but that heavy metal concentration remains stagnant. It's not going high because it's because of geological activities. Often we see release of heavy metals in sweet water bodies or water bodies in general. So we became interested in what is happening, why it is not increasing, because it should increase. Even if you have dumping of heavy metals, we do not see that we have increase in heavy metal concentration in this water. Where these heavy metals are going, they are not going into the soil. Where are they going? So what we did, we looked into the microbiome of the water, and again, as we observed that we have lot of sponge species growing in those waters, so we also checked the microbiome profile of the sponge. So it's the sponge microbiome that we analyzed this time. And here it is the water versus sponge. You see there is a distinct difference between the microbiome. While in water, in Sundarban area, it is dominated by proteobacteria. In sponge, we do see high abundance of actinobacteria. It's a completely different scenario. And when we looked at the diversity, this is again the alpha diversity with a different way of presentation. You see this blue one is always the water from different pond system, and you can see the diversity is higher in water compared to sponge. So the in sponge we have selected enrichment of certain organisms, and when we looked at the microbiome that is present in the sponge and in water bodies, again we see that there is a distinct difference between the composition of microbiome. So we propose that the water bodies and the sponge species harbor different microbial population, and secondly, diversity is higher in water bodies compared to the sponge species. Now the question is why. The next thing we did, we checked heavy metal concentration in sponge and water bodies. And surprisingly, we did see that in sponge species, we have much higher concentration of heavy metals compared to the free water or the water bodies where these pond species are residing. Okay, and when we we moved on. We looked into all the organisms that are present in spawn species. We used a modern analysis technique by from our whole genome data, metagenome data, where we looked into using pie crust. We actually looked into the different functional genes that are present in sponge metagenome and also water metagenome, and that predicted functional gene analysis actually allowed us to identify genes that are present in sponge and in water, which are related to heavy metal resistance, remediation, so on and so forth. And here you can see in sponge, we have high abundance of different uh, heavy metal related genes. Whereas in water, compared to this, all the green ones are highly abundant. In water, you see only this bluish tinge. So it's less abundant. The violet ones are less abundant. Okay, so in water, we have less heavy metal resistant genes or mobilization genes are present in the microbes that are present in water samples, whereas in sponge we have high abundance of these genes are present, leading to propose us that the organisms that are enriched in the sponge microbiome, they are having all these genes within them, and those genes are allowing the sponge to show resistance towards heavy metal and also to accumulate heavy metals on the surface of these microbes. And so we propose that bioaccumulation of heavy metals by different spawn species offer a natural cleaning process. So if you grow the sponges and then if you take out those sponges, you are actually filtering the heavy metals out of the water, and you can actually keep your water clean, having less concentration of different kinds of heavy metal. So with this, I would like to thank all of you for your patient hearing, and I'm open to any question if you have.
Thank you, sir. Any questions from the audience?
Uh, Good afternoon, sir. So
Hi.
I have a question.
Yes, please.
Sir, you said that the Sundarbans, there's a lot of toxic metal deposition in the Sundarbans. Why do you think the toxic metals deposition in Sundarbans, we need to address that? Because it's a World Heritage Site, or because we also know that the Sundarbans, like they say that London has a gate, and for that Kolkata has Sundarbans. So the high tides and the low tides, it's the Sundarbans that protect us from that. So, do you think this toxic metals that we are talking about, and we're talking about the rice plantation in high saline conditions, and these microorganisms which have the ability to form biofilm in these conditions also, how do you think all this can affect lives of people, and how do you think these microorganisms can also improve their standard of living?
Yeah.
Because this recent report have suggested that most vulnerable is women and children is without bias, gender with gender biasness, and they have studies has also come out that it's only through biotechnology or microbiology that we can uplift the entire community of Sundarbans. So, how do you justify this statement?
Yeah, so I think your last statement is really important. And actually, the problem is when we dispose our wastes, even the city of Kolkata, we normally do not process them. So that's one of the issues. And also, there are other aspects like agricultural runoff from different industrial runoff. Unless we process them, what we are doing, we are actually dumping different kinds of pollutants, not only heavy metals, different kinds of pollutants to our riverine system, and eventually these pollutants are ended up in Sundarban. So it was for a long time people have been looking at how these pollutants are distributed in different parts of Sundarban. Lately people started realizing that these dumping can create changes in the biodiversity. Now see, let me give you one example. So as you already correctly mentioned that Sundarban is protecting us. Now one of the important features of Sundarban is the biocementing, the soil system. So the soil system, the biocementing, if the soil becomes weak, what will happen whenever we have another isla or something, a portion of Sundarban will go out. Now that biocementing is very important. And microbes, there are microbes like planktomycetes, for example, they contribute towards this biocementing process. They actually help in biocementing, keeping the biocementing okay, so that the plants can stand rigid even if you have another island. Now coming to what is happening if you increase the pollution, so these good microbes are dying. They are, so that is what we have shown. So basically when you increase the pollution, a portion of microbes that are good, that are really helpful for us, they are dying. Only the one that can deal with those pollutants will survive. So that's why when you see from our data, when you increase the pollution, you have you have lowering of diversity. So that is where the the important lies, why this kind of study is important, to show that we are actually destroying our environment. It will be a long term, but this is important. Now there is another application part. When this is happening, you have the opportunity to grab those organisms that can deal with those pollutants, and you can take them out. You can use them in, through biotechnology, you can use them in agricultural practice. So that is how people are considering this entire aspect, and that is what we also did. Now for example, this sweet water system, I can tell you, we have been collecting water samples from different pond systems in Chagor. Many people actually take water from those pond systems for their household use. So if you have lot of pollution or heavy metal pollution, for example, we still don't know how arsenic is being uh, abundant in Gangetic Plain. It's a natural source. Now, natural leaching is allowing arsenic to come out and released into water bodies. Now, if you have a system where you can really devise something biotechnologically to take out that arsenic from the water, this will be fantastic in near future. Then that household use of water will be okay. You can use it because you can reduce the concentration of heavy metal in that water body. Okay, so that will eventually help the villagers, for example, who are using those waters, because when you use the water, you don't know whether you have any pollutants or heavy metal, because by just looking at the water, you cannot you don't know whether you have any pollutants in that or not. And also there is little opportunity to really in situ measure them. There is no possibility so far in India that you can go and check it in 10 minutes. So these kind of studies are actually offering possibilities that maybe we can adapt this. Let's say you grow some sponge in water bodies and collect all the heavy metals through the sponge and take the sponge out, so that the water body will remain uh, not heavily populated with different kinds of heavy metal. So I think I tried to answer all of your questions. If you have still some question, you can ask me.
Thank you, sir. No, thanks. All my questions are answered. Thank you.
Sir, I have a question. Follow me this side, sir.
Yeah, please.
Okay, sir. It's really a nice presentation, no doubt about it. Sir, I have missed a portion that is when you were talking about arsenic resistant plant and you have inoculated bacillus species. So where you have inoculated it, in the soil or in the root of the plant?
So there are different kinds of treatments we go for. So here it was on soil. Okay, so basically,
and
it's not arsenic resistant plants. So the seeds were selected in order to, I mean, seeds were selected so that they are sensitive to arsenic or stress. So that's why we use IAR 64, because it is showing not much resistance to high salinity. But what we did see in our data that so these organisms are actually collecting all the heavy metals on their membrane surface when you inoculate them, and they get associated with the plant root, thereby allowing less arsenic or heavy metal to go into plant bodies, so different parts of the plant. So it's basically lowering, it's not really allowing the heavy metal to reach the plant tissue. So that is what is happening.
okay, okay, and that's why you get the arsenic in the membrane of Bacillus species,
Yes,
which you
yes,
have
yes, yes.
actually inoculated.
Yeah,
Okay, thank you, sir. Thank you.
welcome.
Thank you, Dr. Ghosh, for your insightful talk and your valuable time with us today. Your presentation was indeed informative, and I hope our participants have gained a wealth of knowledge from it. Thank you, sir, once again.
Thank you all.
The next lecture will be from 2 p.m. onwards. It will be given by Dr. Ishita Rehman, Assistant Professor, Department of Biotechnology, the Nyotia University. She will be speaking on the cancer code. So please join with the link that will be provided in the group. We can dismiss for lunch now. Thank you.